The next paper is by Dr. Michael Reber and Faiza Robbani. And Michael obtained his PhD in human genetics from the University of Diderot in Paris, France. Subsequently worked as a research associate at the Salk Institute. He came to us in 2018 to join the Donald Johnson Eye Institute at Krembil Research Institute, and is continuing to pursue his work on visual network formation and function, and collaborating with Carmela Tartaglia on a research project. And Faiza Robbani, who's also going to deliver this talk, is a student in Dr. Tartaglia's laboratory, working with Dr. Reber, currently a research study assistant in our center, helping with the recruitment of concussion studies. Thank you. Who is going first? Thank you, Faiza.
Hi, everyone. How's everyone doing? Thank you so much for the warm welcome, Dr. Tatter. So today, Dr. Reber and I will be doing a presentation on our ongoing study called the Oculomotor Function Testing with Virtual Reality in Patients with Acute Concussion. So this is an ongoing study. We're actively still recruiting participants. So today, our goal is to describe the experimental design, and Dr. Reber will go over some preliminary data. So some background information. We do know that concussion can lead to changes in brain function that can present in forms of physical, cognitive, and behavioral changes. Some examples of visual disturbances include double vision, light sensitivity, and blurred vision. So there's also ocular motor eye movement disturbances that we may experience. So before I get into that, I want to talk about some uh, eye movements. So there's saccadic eye movements. So that's rapid eye movements. For example, when we're reading, and it's when the eyes are moving from one point to another in a fast-paced motion. There's also the anti-saccadic movement. So that's when we're forced to look at a visual stimulus in the opposite direction. And there's also the pursuit eye movements, which is the smooth tracking of discrete objects that are moving in an environment. So as mentioned, some common eye movement abnormalities include abnormalities in saccade, smooth pursuits, vergence accommodation, photosensitivity. Over here, we have a picture of a brain, and we can see that the eye movements are controlled by a complex, widespread network in the brain. So now I'm going to shift gears here. So the current study we have, it branches off from a past study conducted by Dr. Tartaglia and Dr. Eisenman, where they evaluated the possibility of using SPS eye movement as a marker for post-concussion symptoms and to observe the recovery of post-concussion symptoms. So in the study, they looked at SPS tasks with individuals who had post-concussion symptoms, and they also looked at their white matter tracks. Over here, we can see there's someone who has, they're facing a computer screen, and they were asked to perform some self-paced saccades within a 40-second interval. They also looked at their white matter tracks. And also in this group, they also compared it with some healthy controls. So as mentioned, so white matter, there's some white matter that involve controlling saccades. So I'm just going to briefly give an overview of 4 main pathways that were looked at in this past study. There's the uncinate fasciculus, which connects the orbital frontal cortex to the anterior temporal lobe. So this region helps the brain prioritize important stimuli, and it's important in the visual learning that's gained when performing multiple saccades. There's also the superior longitudinal fasciculus, which connects the dorsolateral prefrontal cortex in the frontal lobe with the parietal regions. So this part of the brain, it plays a crucial role in the attentional network and helps direct the attention to relevant stimuli in the environment. In the bottom here, we have the cingulum bundle. So that connects the cingulum gyrus with the dorsal lateral prefrontal cortex with structures in the temporal lobe. So this region, it facilitates the integration of memory and emotion. So this is the part of the brain that helps with these antisaccadic tasks. And then lastly, we have the corpus callosum, which connects the left and right hemispheres, and it allows the connection between them. So this helps coordinate the eye movements by enabling both hemispheres to work together. So in this graph here, from that past study, it shows the differences in the self-paced saccades between the individuals with post-concussion symptoms compared to the controls. So at the bottom, it shows the interval. So it's within that 40-second interval, and it's divided into four intervals, so 10 seconds each. And then on the y-axis, we have the number of self-paced saccades each group has completed. So we can see for the first two intervals, we don't see much of a difference between the two groups. But starting the second interval, we can see that the patients, the ones with post-concussion symptoms, they're performing far less self-paced saccades compared to the control group. So these findings allowed us to conduct this current study we have going on in terms of evaluating the use of ocular motor function in predicting the risk for persisting symptoms. So now instead of having patients with post-concussion, we want to recruit patients within the acute phase, so within 7 days of their injury. So now we're going to shift gears back to our current study. So the objectives is to identify the differences in ocular motor function between participants with and without concussion, and to investigate the patterns of change and recovery in those who have had a concussion. The secondary objective is to investigate the association between ocular motor function and their concussion-related symptoms. So we hypothesize that patients with concussion will perform worse on ocular motor function testing compared to the healthy controls. So patients who develop post-concussion symptoms by 3 months will display differences in ocular motor function tests in the acute phase, so within their 7 days of their injury window. And the performance on ocular motor function tests will be associated with their symptom burden. So now I'm just going to briefly give an overview of our inclusion and exclusion criteria. So for the inclusion criteria of our study participants, I'm going to refer to the study participants as the patients who have had a concussion within 7 days. So they are within 18 to 60 years old, and they have had to have their concussion within 7 days of their baseline visit. For healthy controls, same age group, their age, sex, and education match to the study participants. And they're included in this study if they have had no past history of concussion. So the exclusion for both the study participants and the healthy controls are the same. So one example would be history of neurological conditions. They would be excluded. History of past psychiatric or developmental disorders that are severe. And one important one is they're excluded if they have any type of vision disorders that would prevent them from performing self-paced saccadic tasks. So here's a very brief overview of our experimental design. So for study participants, we're recruiting patients from the UHN ER, so either the ER from Toronto Western or Toronto General, as well as a concussion clinic at TRI. So if they consent to be contacted for research purposes, I would approach them and screen them for their eligibility. And once they recruit, they will be seen at 4 different time points. So the very first one would be their baseline visit, in which they're recruited within 7 days of their injury. And during this visit, it is the longest visit. So we'll be doing an intake interview where I ask more questions about their past medical history, more information about how they were injured. They'll also be doing a Rivermead post-concussion symptoms questionnaire and also the VR headset testing. So after the baseline visit, they'll be seen for 3 follow-ups. That's 1 month, 3 month, and 6 months from their baseline visit. These are shorter visits. They'll be doing that same VR headset testing. They'll also be doing the Rivermead and just a follow-up interview just to see if there's any changes in their health history. And for the healthy controls, fairly similar. We'll be recruiting volunteers within the community. If they are eligible, they would be coming in for only 2 visits instead of the 4. So it would be their baseline visit and then 6 months after that. So their intake interview, the Rivermead questionnaire, and the headset testing will be identical to the ones which were performed for the study participants. Now, this is just a very quick overview of their intake interview. I've already briefly mentioned that from the past. So this intake interview takes does take long. It just asks more information about their injury, some demographic information, medication history, and see if there's any changes in their medication as a result of their concussion. And they're also given a Rivermead post-concussion symptoms questionnaire in which they fill out on their own. They rate their symptoms on a severity scale of 0 to 4. And from the current group that we have collected so far, we have 7 participants at the moment, and the most severe symptom experienced by these study participants include light sensitivity, fatigue, and sleep disturbance. And the median of the total severity score is 25. So now I'm going to hand it over to Dr. Reber to talk more about our VR headset and also some preliminary data. Thank you.
Hello, everyone. Thank you, Faiza. Thank you, Charles, for the short and nice introduction. So this is the magic tool. Here it's black. Here you have it in white. That's the one we use because it has eye tracking. And so we designed a whole bunch of VR tests to try to understand what's going on in patient with acute concussion. Those tests were designed with Carmela's team to make sure that we address the correct issues and questions. I must say that I'm not a concussion specialist. I'm a vision specialist, so I don't know anything about concussion. And this is a new field for me, so exciting field. And I want to mention 2 of my people in the lab who are sitting in the middle there, Kyle and Danielle, who've been part of both analyzing the data, but also designing some of those tests. So I'm gonna. So we had a bunch of virtual reality tasks. Some of them were existing tasks like the King David test that we just put into the headset. And that's the order of the task that we designed. And so if I press the button now, the video should start, correct? So there is no video for the first few slides. So these are the King David tests. I don't know, are you using also those 3 steps? Okay, so that's task number 1, number 2, and number 3 for the King David test. There is no number 3 here. Okay, the number 3 is those letters. Number 2 is missing. So sorry, this is number 1 where you have the lines between the letters. Then you have version number 2 where you remove the lines, and then you have compressed letters for the 3rd task. And what we do with these is we measure eye position. So we clearly see like eye movements, reading those lines and those letters. And so with fixation over each letter and then moving to the next one. The MOT revision test. So this task is a smooth pursuit. So here you have just a picture of what it means. The participant do not see this white line. This is just to illustrate the laser coming out of the eye, let's say. And so what the eye tracker collects, type of data that the eye tracker collects. After they've done, so this lasts, if I recall correctly, 30 seconds, I believe. They do tracking for 30 seconds. And then the next tasks are multi-tracking objects. So this means that instead of tracking only one single target, as you just saw, they are going to track multiple targets, 2 of them actually, by fixating on that blue dot in the center. So this requires deployment of spatial attention, and which can be quite challenging and cognitively challenging. And at the end, they need to identify the 2 targets that were queued originally. The third test is a so-called saccade test. So it's a mix of, so it's been designed based on Doug Muno's work at Queen's University in Kingston. He's a specialist of eye saccades or saccades in humans, and he designed a so-called iPast, which is the interleaved pro and anti-saccade task. So basically, you have a mix of saccade and anti-saccade task during, I believe, 20 seconds. So here is an example of a pro saccade. So you have to look at the dots at the, so again, the participant do not see the laser coming out of the eye. So you must stare at the center dot for 500 milliseconds before it triggers the stimulus. And then with your eyes, you need to land on the stimulus to extinguish it. So you've seen probably a mix of pro-saccade, meaning that the center dot is blue and then you have to move towards the yellow stimulus. Or if the center is red, I'm gonna try to play it again. If the center is red, you have to generate an anti-saccade. So you have to move in the opposite direction. And we created some sort of a mask. So here is an anti-saccade, right? We created a mask that you don't see, of course, that when your eyes are landing on this mask, the stimulus goes off. And then the last test is so-called useful field of view test. This test was designed in the late 80s, and we just generated a VR version of this task. It basically evaluates different type of attention and vision speed processing. So the first task relates to vision processing. So you basically need to recognize the object in the center between, in this case, an apple and a pear. And this goes faster and faster and faster. It's typical neuropsychological psychodynamics. So meaning that each time you have a correct answer, it goes faster. Right? So you had a few examples of this. And then the second task is recognizing what's in the center, but also localizing what's in the periphery. So this is called divided attention. So we ask you which one was in the center and where was located the other stimulus in the periphery. And again, this goes faster and faster and faster. Periphery being 30 degrees of visual field. And then you have a third version of it where you have distractors. So you still need to recognize, identify the object in the center, and then localize the object, the second object, the periphery. And again, as you succeed, it goes faster and faster. Faster. If you fail, it slows down. So the idea is really to keep you at your perception level. So we have, for now, we have 7 patients and 2 control. So those are very preliminary data. So for example, for the tracking test, this is typically the type of data that we can collect. So as you can see here, the dotted lines are the target. The black lines are the eye tracking data. This line corresponds to head tracking. So we also record head movement. So here in this particular task, you see that the individual was able to perfectly track the target. We can do a lot of math around that and analyze data and measure tracking correlation. Here is another example where we used to have those two targets, and only one is the trajectory of only one target is represented here, where we ask the patient to focus on this center dot. So here are the eyes movement. So eyes are stable within the center of the screen, which means that the patient was indeed looking at the center of the fixation point, and that's the head movement, and that's the ball tracking. And we look whether this was correct or wrong answer. So this was a win, so it was a correct answer, for a ball speed of 5.6 degrees per second. We can have a dynamic representation also of this. So this is one of the patient. As you can see, the tracking here. So the blue dot is the target, and the red lines are the eyes. And so you can see how the individual actually tracks the target by catching up sometimes. And so we can calculate a lot of also data related to these tracking graph, where we know whether the patient was behind or in front of the target, anticipating or catching up, and so on and so on and so on. So this is just an example of the type of data that we can get with this type of tools. That's the RESACT test. So the one I mentioned that you need to move your eyes to extinguish the target. So you were starting here. That's the fixation point, and the target appeared up here. There is a mask around the target to compensate for eye tracking, accuracy, and precision. So it's basically 2-degree mask around the target. And so you see that here, the participant was able to move straight within that area that would trigger the target, that would shut down the stimulus. And then we have representation of time. So here you have the eye position related to time. These allow us to measure the latency of the response. And here you see that you have saccades. Saccades are represented by dots over here. So you have like saccades that are moving towards the target. This is the vertical axis. So basically eyes are moving up. And this is the corresponding horizontal axis. So you see that there is less movement along the horizontal axis altogether, but most of the movement are in the vertical axis. And we can also, that's another example where that was an anti-saccade. So the goal here is to move away from your target, but as often individuals start or have what we call the false start. So they first go towards the target, and then they are like, no, I have to go in the opposite direction. So, and then they correct and end up here by moving in the opposite direction. Okay, same here, you can measure eye movements, and we can also, I don't show you the graphs for sake of time, but we can collect a lot of data, and you have the speed also of the eye that we can measure. Here is the useful field of view test. So this is basically the display time related to the number of presentations. So 10 presentations. As you have correct answers, it goes faster and faster and faster. So display time decreases. This was the first task with processing of this center stimulus and recognizing the stimulus. So all dots are green, which means that the stimulus were correctly recognized each time. And then you reach the limit of the headset, which is basically 14 milliseconds presentation, and people can do this. And here are the two other tests. So one where you have the eccentric stimulation also without any distractors. And you see here two red dots that are indicating that, or maybe I can see here, center is wrong. So the participant here was unable to clearly identify the center stimulus, but was able to locate the one in the periphery. And this is like the third task also, where you have the distractors, and the same issue actually appears, but this is almost the same graph. So you see that when you fail, the next task is slows down, so the display time is a little bit longer. Again, same concept, putting you at the max of your perception threshold. So we have very preliminary results, and I'm not going into the details for the ones where we don't see anything, but so far King-David test, at least for the tracking now, I don't know what type of data you collect in terms of correctly spelling the letters, if there was an issue there or not. But in terms of eye tracking, this was okay. The MOT revision test, I'll show you an example, that was okay. And the useful field of view test were in the normal range. Now, the RESAT test was kind of interesting, and I show you here data between the patients and the control. There are 2 controls and 7 patients, but for a total of 514 trials for the patient and 187 total trials for the controls. What is interesting here is that the number of total trial that timed out. So the trial timed out when you are unable to fixate the target for 200 milliseconds. So basically, you have to keep your eyes in the center for 500 milliseconds, and then you trigger the stimulus, which is randomly dispersed around in your visual field, 15 degrees. Then you move your eyes, if it's a pro-saccade, you move your eyes onto that target, and then you have to fixate that target for 200 milliseconds, so you extinguish the target. Now, if you are unable to fixate for 200 milliseconds, or if your eyes wander around and are unable to reach the target, after 5 seconds, the stimulus goes off, which is what we called trial timed out. And here you see that in total, there are many more trial timed out in patients compared to control. And the major difference actually occurs, surprisingly, in the pro-saccade and not in the anti-saccade. I assumed originally that people would make many more mistakes by doing false starts, so going towards the saccade, towards the stimulus when they are asked to move away from the stimulus. And we don't see too much of this. However, what we see is that they are unable to move and keep their eyes fixated on the stimulus for 200 milliseconds. Now, of course, we have two controls. So if you do the stats with this, it comes up with a very strong z-test, but we are waiting for more of your participant to refine those stats at those numbers. But that was kind of a surprising, but also very interesting aspect. The anti is not different. So healthy control also make mistake by doing false starts. So they move towards the target first and then move away. And we know that this introduces an inhibitory loop, which is essentially controlled by the prefrontal cortex. So whether prefrontal cortex is an area that would be more sensitive to concussion, I mean, that's a question, but there are no difference in the anti-saccade tasks. There we go. So far, I mean, we can't conclude anything. We have interesting data pushing us towards visual behavior that can be affected, but this needs to be refined and confirmed with a larger number of participants. Thank you.
Thanks. This is great. You know, when we first found this thing of saccades being slower, well, less in these, in the patients, we did tons of testing. And like the results here, even though they're preliminary, it seems like when they don't do that badly when they can be slow or stuff like that, but it's just like these basic things. So it'd be really interesting to see in, this kind of analysis, if you can actually break down what it is. Is it they can't fixate? Like we found they actually slow down. I don't know if they get tired or, but maybe over time also you can do the analysis.
I fully agree. I mean, these are preliminary data and we haven't looked at, within the set of data, we can also refine and look more into the time they took to move towards the target, the wandering time. Do they have straight trajectories or do they move around or things like this? We haven't looked at that, but for sure, thanks to the eye tracking, we can measure that. Now, I fully agree. I believe they do quite well when it's too easy or even it's easy. And then when it becomes cognitively challenging, then this is when I believe you can reveal some differences, And the paper that you published previously, I looked it up, but I didn't remember that graph that Faiza showed. But to me, it's pretty stunning that, things appear after the third and fourth trial. 1 and 2, you don't see statistical differences, So they have to be, it seems to me that if you want to use some of those tests to diagnose or screen for concussion, you have to design tests that are challenging, even for us, like healthy individuals who do not sustain concussion or whatsoever, because otherwise on the easy tasks, they do quite well.
But actually, I think your data shows that even in the easy tasks, one of the issues that we have with concussion patients is many of them complain of fatigue. And I actually think some of that fatigue means that they are using more brain recruiting areas of the brain that may not be as efficient. And so even things like a saccade task, which to me is one, like, of course, you see patients who lose that, but it's an easier task, right?
Yeah.
And yet you can still see, and even now with all this complicated data, you still see that saccades show a difference. And so I just wonder if there's some basic thing in the system that doesn't allow them to just do these. And so that's why they feel so tired, because it's basic things, not complicated things.
Yeah, but I think we need to look at, the brain areas that are involved in saccade control, and then look where those brain areas are located and how. And that was one of the questions I wanted to ask previously to the other speakers is, are there any, as I said, I'm not a concussion specialist, but are there any mechanistic hypotheses about, accommodation deficits, which are major symptoms, related to which brain area or which pathway or which connectivity system would be involved and relevant to the concussion itself? What does it mean? Is it because you have, if I understand correctly, you have a rotational concussion also, right? So is it involving only the area that are going to hit the skull, or are areas where you have like the highest displacement? I don't know, like more midbrain areas. Are they moving a lot in terms of distance and force? Maybe we should look at this first and have some information about which areas could be physically damaged or.
Yeah, there is some data that rotational acceleration is one of those areas, but brain stems are difficult to image properly.
Yeah.
But thank you for
Have to move on. Thank you very much, Michael
Thank you.
and Faiza.